So for this audio lecture, I want to provide you an introduction to parasitology. So what are parasites? We're going to look at relationships between organisms. And then we're going to do an overview of different types of parasites. Next week's lecture will go into more details on the different types of parasites. So again, today's lecture is more of an introduction. Also, we'll talk about some of the different terms that are used in parasitology. So what is a parasite? So when we look at a parasite, it all comes down to relationships. The relationships that organisms have between each other. And for a parasite, for our purposes, our definition is going to be a general one, where a parasite at some point in its life cycle causes damage to its host. Okay, and as we go through our lecture today, we'll kind of see how this is not as clear-cut as one would think it is. But again, this idea that there's some kind of harmful effect that's going to occur to one of the organisms that are in a relationship. So next what we're going to do is talk about some different types of relationships. So the term symbiosis was coined back in 1879, and it means living together. And so symbiosis is looking at two organisms from different species and how they're interacting with each other. And these interactions can be beneficial, they could have no effect, or it could be a harmful effect. So this table does a nice job of summarizing the different types of symbiotic relationships. If we look at the table, we'll notice that we have a column for population A and a column for population B. So keep in mind that when we're talking about a population, we could be talking about a single organism or species and how it interacts with another organism or species. Okay, and so the relationships we're going to focus on are mutualistic, commensal, and parasitic. So with the mutualistic relationship, as you can see from the table, both population A and population B are going to benefit from those interactions with each other. For commensal, we're going to have one population is going to benefit, but the other one is unaffected. So they're not harmed, but they don't benefit either. And then with parasitism, this is where we're going to have one benefiting, one population is going to benefit, where the other one actually is harmed. So it comes at a cost. And for population B, for the host, it's not beneficial. So we're going to look at some examples of these three different types of symbiotic relationships. So again, with a mutualistic relationship, this is where both species are going to benefit from this interactions. And there's lots of examples on the planet of mutualistic relationships. So we could look at something like insects and plants and how insects help to pollinate plants as they're feeding from nectar. So plants are providing nectar for the insect picks up pollen, moves to the next flower, and actually will pollinate that flower for them, allowing then for the seeds to become fertilized and then development of fruit. If we, as a microbiologist, if we look at us humans, we have our gut microbes, and our gut microbes are going to be important for digesting. So when we eat, we are not just feeding ourselves, we're also feeding our gut microbes. Those gut microbes are going to help break down that food. They're also going to produce vitamins and hormones to help keep us healthy. Now, the gut microbes also benefit from this because they now have a house. They have somewhere to dwell and live. They have to compete with other gut microbes, but they don't have to compete in the outdoor. They have their host. It's also beneficial because you're feeding them, right? So they don't have to go and fight and compete for those resources. Okay. Now, when we look at a commensal relationship, this is where one organism is going to benefit, but the other one is not harmed, but does not benefit from the relationship. So if we think of a kind of a larger scale example, if a spider is building a web on a plant, the plant's not going to benefit by having that web there, but they're not harmed either by the web. But the spider's going to benefit because now they have a web, they can catch food, they can eat. Okay. If we look on a smaller scale, a microscopic scale, if we looked at our skin, we have Staphylococcus epidermis on our skin. It's a pretty common skin bacteria. Doesn't harm us. For this purpose, we'll say it doesn't really help us either. And it actually just feeds off the dead skin of our dead skin cells. So I kind of hesitated saying that there's no benefit because by having Staphylococcus epidermis on our skin, it actually protects us against picking up potential pathogens. So again, sometimes this one's commensal relationships are a little bit blurry because if we delve in deeper, then we might see that there are some benefits that occur. With a parasitic relationship, this is where one organism is going to benefit and the other one is actually harmed. Okay. And so in the microbial world, there's lots of examples. And so we have numerous microbes that can actually cause disease. So these disease-causing microbes would be example of parasitic. Also, if we talk about viruses, viruses are known as obligate intracellular parasites. So they're required to be inside a host cell, and they're parasitic. They cause damage. Again, this is kind of an oversimplification because there is that potential that even these disease-causing organisms aren't always disease-causing, that they could be beneficial in certain snapshots, in certain parts of their life cycle, or for certain hosts, certain organisms. Okay. So again, we'll look a little bit more about this idea of parasites and how to define them in a moment. What I want you to do is have another analogy to think about these different relationships. So, as college students, you've probably had roommates or you've had siblings and you've had to share with your siblings. And so if we think about those relationships we have with people, they can also fall into these different symbiotic relationships, and they make a nice analogy for you to really understand these different types of relationships. So for a mutualistic relationship, this is where your roommate wears the same size shoes as you both have the same taste, and so instantly your wardrobe has increased, and so you have more clothes to pick from, more shoes to pick from. And they also are very respectful, and so they always make sure they return anything washed and no rips or tears or anything like that. So a very mutualistic relationship, and the sharing goes both ways, right? For a commensal relationship, this is where maybe your roommate doesn't have the same taste as you, but they also like your clothes. So they're going to borrow your clothes. But again, they're going to return them washed and not ripped or anything. And so you don't really benefit because you aren't going to borrow their clothes, but they're going to benefit because they're. So that would be a commensal relationship, where if we take this analogy and we move it into a parasitic relationship, this is where the roommate takes your clothes, borrows them, doesn't return them clean, they've gone missing, they've got rips, so now you got to buy new shoes because they broke the heel in the shoe. So again, hopefully this analogy kind of helps you think about the different types of relationships. So as far as the exam, you should have a good working definition of these three different types of symbiotic relationships. And be able to provide examples of them. And we can think about those examples in our roommate analogy, or we can think about those examples in a larger scale as far as like plants and insects, or we can think of them in a microbial point of view, a smaller scale. So when we're looking to identify which organisms are parasitic, it's really these harmful effects that can happen to the host. That is a general way in order to define them. So usually the emphasis is on one species living at the expense of another. Okay. Now we want to keep in mind that for some, the definition of parasites is by the mode in which they cause harm. So for some parasites, they are causing harm because of being metabolically dependent upon their host. So these are autotrophs. So they're requiring a food source from their host. And so they now are competing for nutrients from the host. And so the host isn't going to have enough nutrients to support themselves. And so that's where the negative effect happens to the host, is because of that stealing of nutrients. And again, so some parasitologists define a parasite because of the metabolic dependency. The problem with just using that as a criteria for whether something is parasitic or not is that some parasites compete for habitat. And so they have a habitat preference, and because they're in that habitat preference, they're parasitic. And so if we move them to a different habitat, they wouldn't be parasitic. So those are the two main modes in which hosts have harmful effects occur to them: metabolic and habitat. And so if we just use one of these criteria, we miss some organisms that could be parasites. And so having a more general definition may be better, because then you're catching more. So if we look at the Webster's Third New International Dictionary and look at the definition, we'll see there's lots of different pieces to it. So one, we have organisms that are living in or on. And so we'll talk about this idea that we've talked about these organisms living inside, so causing disease to humans inside of us. But parasites can also live on another living organism. So they obtain in part or all of its organic nutrient, right? So again, this idea of metabolically dependent, using the other living organisms' resources, their food source, commonly exhibiting some degree of adaptive structural modification. And so this adaptive structural modification is going to be important in how we start to identify different types of parasites, and will be very helpful in the lab when we go in the midpoint of the lab this semester. We'll have you look at some different slides, and it's those structural modifications, those different cellular structures that help us to identify the different parasites for the different species that they are. And then the last piece of this definition, causing some degree of real damage to its host. So going back to this harmful effect in which a parasite, an organism, can have on its host. So as I mentioned, this idea of what is a parasite becomes a little bit tricky, and we can either have a narrow kind of approach to defining, and that actually excludes a number of them, or we can have a more general approach at defining what a parasite is, and then we kind of capture almost every single organism. So there's really no distinct ecology, function, evolution, or physiology that distinguishes all parasites from non-parasites. And so we really need to take a whole view of the life cycle of an organism and examine each part of that life cycle to determine whether an organism is a parasite or not. And so kind of lastly, concluding in this, the reality is maybe we all are parasites at some points in our lives, and kind of wink with that. And that is truth, because, if we think about us as in development as a fetus, we are feeding off of our host. We are relying upon our mother to provide us with nutrients, and that comes at a huge cost, and in many cases in a harmful way to that host. Okay, so we're going to look at some of the key terms in parasitology. So these are terms that we'll be using throughout this semester. So the idea of a host. So host is where the parasite is going to be living in. And usually that host refers to when it's exhibiting its parasitic behavior. We can have obligate parasites. So for obligate parasites, these ones are pretty easy to define as parasites. They require this parasitic behavior in order for them to survive. Where facultative parasites are kind of like you got to catch them in the right environment. You got to catch them in the right host, in the right organism, in order them to be viewed as parasitic. Okay. And then if we look at where the parasite is found, we can have ectoparasites or endoparasites. So ectoparasites are going to be on the outside of the host, and endoparasite is going to be within the host. Our next column has some different organisms that are known as parasites that we'll be covering in this course. So we have Protozoan. Those are unicellular eukaryotic cells. We'll talk a little bit more about them later. Helminths, also known as parasitic worms. Under the Helminth category, we have nematodes, cestodes, trematodes. So these are all different types of worms, and so we'll talk more about those in a little bit. And then we have vectors. So vectors are organisms that allow for transmission of the parasite to a host. And so a common vector are arthropods. So we would think of those as insects. Okay, so these are some of the different key terminologies that we'll be talking about throughout this semester. So when we look at parasites, parasites can infect humans, but they can infect other organisms. So even plants can have parasites. Our focus this semester will be in humans, but we will also talk about other organisms, ones that relate to veterinary medicine. So the CAPC lab actually looks at veterinary medicine data in dogs and cats. But you might find that if veterinary medicine is of interest of you, that you select a parasite that's relevant in veterinary medicine to do your paper on. We'll also be looking at the technique of fecal flotation that can be done in humans, but the data we'll be looking at is in horses, equines. So again, for a lot of times we're talking about in humans, but keep in mind that there are parasites in other organisms also. So when we look at parasites, they can be classified in different ways based off of characteristics that they have, and also parasites that infect humans have various life cycles. So there's several different ways in which to teach parasitology. One of them is very life cycle driven and characteristic driven. And so memorize your life cycles, regurgitate life cycles, memorize characterizations, and regurgitate that. So I like to take a more approach of balancing that with the concepts, how this relates to public health, how this relates, how parasites are looked at in the labs, what are the techniques that are used, and also using primary literature of what current research is on these different diseases that are caused by parasites. So we will talk about life cycles for these different organisms, but we'll be talking about them in a more general approach. Obviously, for the parasite that you pick for your paper, you should talk about their life cycle. You should include a figure on their life cycle. So that information is very important, but there are hundreds of thousands of different parasites, and so in no way we are just touching the tip of the iceberg in the parasites that we're going to talk about. And so their life cycles are so varied depending upon what parasite you're talking about. So we will spend our efforts on learning other information than just memorizing life cycles. So as I mentioned for protozoans, these are unicellular, one-celled eukaryotic organisms. And so for the most part, the general accepted method of classification is based on how they move. So what cell structure do they have that allows them to move? And for some of them, they don't. So there's a fourth category. So when we look at, so for protozoans, they can be classified into 4 different groups. Sarcodinia, which are the amoebas. So these are going to have pseudopodium that allow for their movement. And in our next figure, I'll show you some pictures of these different groups. The mastigophorias are also known as flagellates. So Giardia is an example of this. You'll be looking at Giardia in your Cap C lab. So again, flagellates, Giardia, they're going to have flagella, and those flagella allow them to move. The Ciliphoria or ciliates have cilia that line them. You might have seen like a paramecium before. So a paramecium is going to have cilia all over the surface, and that allows it to move. Just be aware that some ciliates are not motile. They actually use their cilia to move material into their food groove. And so for the Ciliphorias, the presence of cilia does not always mean that they're motile. The Sporozoa are our last group, and this is the miscellaneous group. And so these are the ones that are not motile. So they don't have movement based on having a structure like pseudopodium, flagella, or cilia. So this figure just shows you some different examples of what some of these protozoans would look like. So in panel A, our paramecium would be a ciliate. In panel B, our amoeba is going to be our sardinia. And then panel C, this would be an example of a flagellate. So for the helminths, the helminths are going to be our parasitic worms. So these are eukaryotic also, but they're multicellular. Okay, and if you've ever had a pet or maybe a child that has had a parasitic worm infection, maybe they had tapeworms or pinworms or roundworms, you know that you can see them without the use of a microscope or a magnifying glass. So at the adult stage, these can be quite large. So like protozoans, they can have free-living parts of their life cycle, but they can also have parasitic stages. Okay. Now, it's interesting to note that with Helminths in their adult form, they cannot multiply and increase in numbers. So for their life cycles, they have an egg stage, a larval stage, and then the adult stage. In the adult stage, there'll be eggs that are produced, the eggs are fertilized, those eggs usually get shed, and then they get picked up. And this is why dogs, pets, dogs, and small children can become infected with them, because when they're playing outside, they might accidentally touch some of these eggs that are microscopic, that you would need a microscope in order to see. So it's not that they realize they're touching it. And then because pets and kids tend to put hands or paws, lick their paws, they're then ingesting that egg. That egg then gets into the GI tract. Those eggs then kind of hatch. That allows them to become larvae. Those larvae are released and they become adults. And the cycle continues. Okay. So many of the hemolymph are very treatable, and we'll talk about treatment and controlling of parasites later on in this semester. So when we look at hemolymph, they can be broken into 3 main groups of hemolymph. Okay, and these are ones that affect humans. So our flatworms, or platyhelminths, those can be further broken down into trematodes, which are known as our flukes, or cestodes, which are known as tapeworms. So with flatworms, these have a flat appearance. For trematodes or flukes, they're not segmented. So if you've ever seen a flounder, flounder is a fish that kind of skirts along the bottom of the ocean. It has a very flat appearance to it. So flukes almost look like flounders, but much smaller. Okay. Cestodes, tapeworms, these have segmented pieces to them. And so again, you might have seen these if you have a dog or a small child that's had tapeworms, and you can see it looks like little pieces of tape. And so one of those segments, when it's mature, has both male and female sex organs, so that they're hermaphrodites, and so that they can reproduce and create, eggs and then adult. And so those segments can actually, one segment can be sufficient to then cause an infestation. Our next group of Helminths are horny-headed worms. So these actually have spikes on their heads, and those spikes can allow them to attach to the intestinal wall. So they're found in the GI tract. Okay. For our roundworms, our nematodes, these are going to have a round appearance. So again, flatworms, flat, roundworms, round. And the roundworms can be non-segmented. Some people do consider the segmented worms, like annelids, or like earthworms, to be part of a hemolyth. Other people put them into different categories. So as far as this course, we might end up talking about leeches. We don't usually, and I would think, given the format of this course this summer, we probably won't be talking about leeches. If I taught the course during like fall or spring, I might be able to sneak that in somehow in the paper or something. I would entertain someone doing a paper on leeches if they wanted to. Okay, so going back to roundworms, these can be, the adult forms can be found in the GI tract, blood, lymphatic system, and also in subcutaneous tissues. But the immature, that larval stage, can be found in other parts of the body too. So the Helminths are all great examples of endoparasites, right? So we talked about them living inside the GI tract, but we also have ectoparasites. So ectoparasites are going to be ones living on the surface. And so as I mentioned before when we were talking about the different terms, arthropods would be an example of ectoparasites. So something like a mosquito, right? Or ticks and fleas and lice and mites. These all can be parasitic. They can cause disease. If we look at mosquitoes and ticks and fleas, those can transmit through a bite the parasite. So even they may not be parasitic themselves. They may not cause disease themselves, but they will transmit the parasite that will cause the disease. Okay, so they're really important as vectors for the life cycle of many of our parasites. So one of the other things I would like you to do is to listen to Ed Young's TED Talk entitled Zombie Roaches and Other Parasite Tales. Each year it's becoming older and older. So it was originally filmed back in 2014. But some of you may have heard of Ed Young. He's a scientific writer. He's written several books, and he's had some different podcasts available, and so he's wonderful to watch. I love TED Talks, but Ed Young, he's amazing in how he delivers the scientific information, but makes it relevant and interesting. So in this TED Talk, he talks about some of the different parasitic relationships that parasites have with their host and how they can change their behavior. And I'm in agreement with him in that we can find all these examples in other organisms in how parasites can change the behavior of their host. So for one example, there is a worm that will infect shrimp. So the shrimp normally are very isolated, but when they're infected with this parasite, this worm, they start to cluster together. They also start to have a pink color to them. And so this pink color makes it easier for a flamingo to see them. Plus, they're together in like a little school. And so it's the flamingo can catch many of them because they're all hanging out together. Well, the flamingo is the host for the worm. So in order for the worm to complete its life cycle, it needs to get into a flamingo. And so by using this intermediate host, by using the shrimp, by changing the shrimp's behavior, it's going to be more likely to get into its host, the flamingo, right? So you've probably been to like the zoo or somewhere and you saw flamingos and you're like, oh, look, that's so cool, they're eating the shrimp. And you never realize it's all driven by a worm, right? So he gives a number of other examples of these changes of behavior because of parasites. And so for humans, we're really hesitant to give power to parasites in some of our behavior. So one of the things he does talk about is toxoplasmosis. So toxo is a parasite, and it's found in mice. We know it can infect humans, and it needs cats also. So it needs cats in order to survive. And so mice that are infected with toxo do not fear cats the same as mice that are not infected with toxo, right? This lack of fear of cats allows for the toxo, the parasite, to get to its host, cats. So one of the studies that Ed Young talks about is where they have looked at cat ladies, right? So affectionately, these individuals who have this kind of need in order to have more cats. So they have looked at those individuals that have a high number of cats, or affectionately known as cat ladies, and they have toxo antibodies. So what that means is if they have toxo antibodies, they've been exposed to toxo at some point. Okay, it doesn't mean that there's an active infection. So toxo goes into the brain, and so obviously we don't want to just biopsy people's brains just because they like cats. So looking at antibodies is a very non-invasive way in order to do that. So they found that individuals that would be considered cat ladies tend to have a higher incidence of having antibodies against toxo. So their love for cats, is that actually because they love cats, or is that because the toxo is telling them to like cats so the toxo can get into their host, okay, to complete their life cycle? So Ed Young does a very nice job of presenting some of these interesting. So I really just want you to think about, how do you view parasites now? And hopefully at the end of the semester, maybe you'll view them slightly different. Maybe some of those quirky behaviors that you have, maybe they're driven by parasites. Food for thought. So for these slides, a number of resources were used. So there's an OpenStax microbiology textbook. So if you don't have a micro textbook, it's free. Go to it. And it's not that textbook will be used for the whole semester, because a lot of that textbook covers bacteria and viruses, and that's not going to be our focus this semester. Our focuses are going to be on the protozoans and the hemolyths and ectoparasites. But any microbiology textbook definitely touches upon that, and that would be great for background information. The CDC also has a website on parasites. They also have websites on individual parasites, but the link that's provided here brings you to the general page. And then obviously you can search for different parasites. Like if you're doing your paper, you can look at what are some of the different parasites that there's information available for. And then we also have Ed Young's talk. These resources will be used throughout the semester, especially the CDC and the OpenStax textbook, so they're good reference points. There are a couple other resources that will be used during the semester, and I will always provide links to those sources so that you can go to them, because we don't have a traditional textbook. Okay, So hope you enjoyed this lecture, and I will talk to you later.